Hello viewers, welcome. This is Disease Reversal and Mitigation Studio. I'm your host, Fred Mushiri. We have a very important topic to discuss today on the mystery surrounding the Spanish flu virus. And I want to take you to the year 1918 when we had a pandemic that was called the Spanish flu. And you could still be asking yourself, why was it called the Spanish flu? We'll come to explain that. But it claimed, at least according to scientific estimates, 50 million deaths, and some other sources were putting it to be around 100 million. This could have been the deadliest outbreak in history, claiming about a third of the global population then. So that was in 1918, at the end of the First World War. Now, the pandemic was blamed on a type of virus we call the H1N1 strain that is said to have jumped from birds to humans and have caused a global infection. And it caused mortality, particularly in the healthy young adults. Now, there are three issues here we need to raise eyebrows. One is that it had jumped from birds to humans. We do not have any evidence or information that there is in any way people around that time were interacting with birds. And it is not also reported that birds were dying because of any disease around the time we had this pandemic. So that's number one. Number two, when we do not have diseased animals, it is unlikely to have a pandemic because the animals have to be sick first before they're able to spread the disease to humans. Now, the other issue we need to raise eyebrows for is that it was a global infection. For infection to be global, it must have affected all the continents. And if it affected all the continents, it means the infectious agent had the energy and ability to survive in all those world environments. Now, this is not true with microorganisms because they are tiny and they do not have enough energy to move from one continent to another, given that we have varied climatic conditions in the different continents we have on the planet. So it is unlikely that a microorganism will cause a global pandemic. A natural microorganism cannot cause a global pandemic. It doesn't have the energy. Consider also that even the larger organisms, such as elephants, do not have the ability to occupy any climatic zone in the world, despite the fact that they have all the energy. The other issue we have to doubt is that the disease affected mostly the healthy young adults. It is unusual for any infectious disease to affect people who have the largest share of immunity. When infectious diseases come, they'll get rid of people who have weak immunity, for example, the sick, the elderly, the children, but not the healthy young adults. Okay? Now, this claim could have been placed here because we know the young adults participated in the First World War and they were killed in large numbers. That can only be the only reason. So, we also told that the virus emerged in the US, despite the fact that it's called the Spanish flu, but we come to explain why it was thus called. It emerged in the US and then spread across the other continents. So why was it called the Spanish flu? The 1918 influenza pandemic was nicknamed the Spanish flu because Spain was neutral, because it was not censoring information from alternative sources. So other countries were doubting and they were questioning the information given by mainstream science, but Spanish was not. So it could have been mocked. That is, Spain could have been mocked by the flu being associated or called after that nation, the Spanish flu. And the idea to do with restriction of freedom to speak or to question mainstream science was witnessed in 2019 and 2020, 6 years ago, when we had another pandemic and no one was supposed to give alternative information. So even people who are giving diverging views on the pandemic of 2019, they were being ostracized from social platforms. So we had the same thing in 1918. Now, let's consult divine intelligence to know the order of pestilences. We are going to read Matthew 24, verse 7. It says, "For nation shall rise against nation, and kingdom against kingdom. And there shall be famines and pestilences and earthquakes in diverse places." And now, because we want to understand the mystery surrounding the Spanish flu, let's consult what the Bible says concerning pestilences in the times of the end. Now, consider the sequence given by divine intelligence here. We have events or disasters of human creation. We have the war, we have the famine, and then the pestilence. And then we have the earthquakes. So eventually, the events of human creation culminate with a natural occurrence which just comes to heap up to the disorder that is already there. But one important thing that we need to learn from this verse is the event that is preceding prepares the ground for the subsequent event. So the war resorts to famine, and famine resorts to pestilences. And then after those human disasters, we have a natural event, which is the earthquake. The second thing is the events have to be in diverse places, including the pestilence. So the meaning of the phrase diverse places here is distributed widely. And we've said it is unlikely for microorganisms, given their tiny nature, and they do not have enough energy to be able to spread diseases across all the climatic zones we have in the world. So a pestilence has to be in diverse places. It has to be across the globe. And infectious agents do not cause global pandemics. So if anybody says that the Spanish flu was as a result of an infectious agent and that the flu was global, then that one defies what we call the natural order of things. It also defies the sequence given by the Bible, that the pestilence has to be global or in diverse places. And we know that microorganisms do not cause epidemics that are global because they are tiny and they cannot adapt to different environments. Now, we get the same confirmation from other inspired writings. For example, first volume of Testimonies 268. The order starts with human disasters, that is, disasters of human origin, strife, war, bloodshed, which lead to famine and then to pestilence. So we are told it is the war that caused the famine, and the famine or want caused the pestilence. So that's the order of events according to the advice given in the inspired writing. Okay, now let's go to the actual event. In 1914, we had the beginning of the First World War, and there are reasons that are given as to how the war started. It was triggered by the assassination in 1914 of the Austro-Hungarian Archduke Franz Ferdinand by a Serbian nationalist. And that event escalated from a regional conflict into a global war. So when the war started, it created dire circumstances that predisposed the population to diseases. So to start with, we had mass casualties. So 20 million deaths from the military died. So this could be the same figure that has been summed up with the 50 million that died because of the infectious agent. And we have seen this in the past. In 2019, 2020, anybody who died of any disease was classified as having died from the pandemic. So the same applied during the Spanish flu. Anybody who was dying during the war, regardless of the cause, were counted as to include the number that was eventually tallied as deaths from the pandemic. So we also had soldiers who had very noisome and dirty wounds because soldiers were fighting in trenches. And if they were injured, the wounds were likely to be infected because they were dirty. If you get a septic wound, that is a wound that is dirty and has an infectious agent, it is likely to spread the infection in the entire body. And the mode of death of somebody who has an infection in the blood is by a respiratory failure. So they die because of failure to breathe properly. They have a difficulty in breathing. And because the cases that had the Spanish flu were said to have died of respiratory failure, now you expect that one to be the case to anyone who has died of any infection that has gone into the blood. We also had changing role of women, where women were to take up the jobs that men were doing. And when women who are responsible for keeping the compound and the house clean go and take up the jobs in the industries, then you can be sure the children that were left were likely to pick diseases as a result of poor sanitation and malnutrition, because some were not getting to be breastfed because mothers were not anywhere at home. So diseases of malnutrition and poor sanitation also arose because women were not to be found anymore at home. They were supposed to take up the jobs that men were doing. And then we had the psychological stress that was cutting across all groups of people because of the war. And this must have contributed to low immunity among the population. And then there was famine, there was inflation. Anytime we have a war, there is always an attempt by the opposing military to sabotage. And the sustenance is the first one to be sabotaged. So they destroy crops, destroy water, and other social amenities that may enable their opponents to have an advantage. So we also had food being taken away to be used by the military because they could not predict when the war would stop. So there was hoarding of food in stores to be used by the soldiers. Then we had use of chemicals which led to air pollution. There was a polluted atmosphere. There were people and animals decomposing on the surface of the earth in all places. So this could have led to the pollution of the atmosphere that could have caused lung infections, and eventually all those cases who died were counted as dying from the Spanish flu. Then we had internally displaced people, and you might have witnessed places where there is war. When people are internally displaced, they are crowded, and if they are crowded, it would mean they scramble for the few sanitary services available. So there will be a lot of accumulation of waste, pollution of the atmosphere, and deaths coming as a result of poor sanitation in their habitats. So all those circumstances prepared the ground for the coming of a pandemic. Unfortunately, this pandemic was called the Spanish flu, and it was attributed to an infectious agent that is said to have caused the disease across the globe. So that infectious agent, they called it a virus. But we want to have a very brief description about what a virus is. Now, getting information from mainstream science itself, viruses are considered to be non-living. And if you hear something is non-living, the opposite of non-living is living. And the opposite of living is dead. So if something is non-living, it is dead. So in other words, viruses are considered to be dead. They lack what we call the cellular structure. They cannot reproduce. They cannot metabolize. It is said that they can do nothing independent of themselves. They rely on a living cell in order to replicate. But we still have a lot of questions. How they replicate, because they are non-living. Now, if you read Genesis chapter 1, verse 24 and verse 28, we have been given the characteristics of living things or living organisms. Number one, they have to reproduce, and number two, they have to move. So those are life characteristics that identify a living organism. So viruses do not reproduce, they do not move, and therefore they are dead. They are non-living. And this is mainstream. This information is coming from mainstream medical books. Now, the only difference here is that the claim is they have a genetic material. Now, we need to know that it's only living organisms that ought to have the molecule of life, the DNA, because this is the one that enables those living organisms to reproduce and to have life characteristics. If something is non-living, it ought not to have the DNA molecule or the molecule of life. DNA comes from components generated by living organisms, not non-living organisms. So it's important to understand that information concerning viruses, how they have been classified. And that is why when you look at the biological classification, it does not include viruses because they are considered to be non-living. Now, with this information in mind, let's move on. We want to uncover the mystery surrounding the Spanish flu epidemic or pandemic of 1918. Now, looking for sources that are credible, you get the following information. That in 1951, that is 33 years later, a young microbiologist called Johann Houghton traveled to Alaska and exhumed a mass grave of the 1918 pandemic victims that were buried in the permafrost. Now, the first issue you have to raise here is this. Now, suppose you are to return to a place 30 years later. There's a lot of change that has occurred on that place. Most likely, you will not be able to identify the place. But interestingly, we are told that Johann Houghton was able to identify a mass grave, and he was sure that this mass grave belonged to victims of the 1918 pandemic, not any other victim. Now that is where the problem starts. Next, we are told that he obtained samples from the bodies, but then he was not able to study the samples because certain study techniques were missing. Certain modalities of studying the samples were missing. And the missing techniques included the electron microscope. The electron microscope was discovered in 1931 by a German physicist, and by the time Dr. Johann Houghton was extracting the samples, this microscope was not developed to the extent it would be able to study conclusively the virus. So there were other techniques John Houghton had to wait for. For example, what we call the PCR. Because PCR is very important in study of microorganisms which are of the size of nanoparticles, okay, so small. So it needs modalities such as PCR and also the electron microscope to be able to study. So he had to wait until later than 1983 because the PCR machine was invented by Carl Mullis in 1983. So he had to wait for it to be developed to the extent it could study his samples. And this takes us all the way to 1997. So in 1996, we are told that Dr. Jeffrey Taubenberg and his team at the Armed Forces Institute of Pathology successfully sequenced fragments of the 1918 virus genome. And you can see this work is being done in a pathology lab belonging to the armed forces. So the question we're asking, why is the military interested in medical research? What is this? What's the relationship between the US Army and medical laboratories? And if you look at one of the papers of 1962, July 15, it had reported that the Army was experimenting with super germs. So we are not surprised if we hear that these samples that were taken from bodies in Alaska were studied or investigated in an armed forces pathology laboratory. You also remember that in 2015, the then president of US, Barack Obama, commissioned 3,000 troops to go to Liberia during the time of the outbreak. And one of the dailies had this caricature of a patient lying in bed, and you have soldiers with guns storming into the sick room. So you wonder what have the boots and the guns have to do with a sick patient who is too weak to wake up from his bed. And this is such event confirm that there's a lot of interest the military has on superbugs and other infectious agents. Okay, maybe possibly because of the biological warfare research that is ongoing in certain laboratories that have been militarized. Now let's go back to the mystery. 79 years later, after Houghton now brings the samples to a military pathology lab to be studied, in 1997, he went back and collected a few more samples in Alaska. That is in 1997, 79 years later. He collected samples in an attempt to get the virus that caused the Spanish flu. Now, because viruses depend on a living tissue, on a living cell to be able to survive, if they can survive, please know they are non-living things. If they are non-living things, do you expect they survive? But he went to a body or a dead cell that has been dead for 79 years and could have been decomposed to find a virus. Now, do you expect a virus to be found in a dead tissue for 79 years? I leave it to you to judge. So, because we do not have the techniques, as I said, such as PCR, and also the electron microscope was not fully advanced, the samples had to wait until 87 years later. That is in 2005, for proper reconstruction to be done. So we are told that these samples were sequenced and about 8 genes were sequenced and reconstructed, and it was reported that that was the virus that caused the 1918 pandemic. So you'll be surprised to learn that the virus that is said to have caused the 1918 pandemic was a reconstructed virus. So it was something of human manufacture. So in 2004, we are told the complete infectious 1918 virus was successfully reconstructed using reverse genetics technology. So that is actually what happened. The virus was reconstructed, and this information is available on the mainstream science. So what is this about sequencing and reconstruction? Now, there's a problem when it comes to sequencing a virus that is RNA based. Note the viruses are classified into two. We have the DNA viruses and we have the RNA viruses. Whether this is true or wrong, we can confirm in a later video, but for the Spanish flu, it is classified under the RNA category, viruses. So there is a problem when it comes to sequencing of RNA viruses or reconstructing them. And I'll tell you where the problem is. Number one, we need a PCR to sequence an RNA particle. A PCR is needed. Now, the problem with the PCR is that it cannot tell you what is in the sample. So it will not tell you whether the sample that has been collected is dead or alive, because it's a machine. It does not have the ability to identify the characteristics of life. So it just quantifies. And the way it quantifies, once it is fed, as you can see on the screen, on your left, once the PCR machine is fed with a DNA or RNA, then it multiplies those sequences up to the number that you have programmed it to multiply. Say, if it's 30 cycles or 35 cycles or 40 cycles, and it will give you as many sequences as you want. So these sequences will be legible. or you'll be able to read them because they are many. But what it does, it multiplies them, but it will not tell you what is in that sample. So those are the challenges of PCR. So meaning you cannot use it to diagnose, to tell somebody that you have this virus. You cannot use PCR to tell somebody you have a COVID, or you have a hepatitis, or you have a Spanish flu, but it cannot identify which virus. But depending on how it has been programmed, it will multiply as many times as you want, whatever you have fed into it. That is why we call it, we call the method used, the working principle of PCR is called molecular photocopying. So it will give you as many copies. It's like a Xerox machine. It will give you as many copies as you have programmed it to give. So the person who discovered the PCR machine, Kary Mullis, had this to say about the PCR. Remember, Kary Mullis was given the Nobel Peace Prize for chemistry because of this discovery. So he is a credible source that you can believe. So the PCR test, he says, is not a diagnostic tool, and it was invented for detailed study of DNA samples. So it will study DNA better, but not RNA. That is why it is also used for determining paternity with the DNA, because once the sample is fed into the PCR, it will make as many copies as possible to be legible or readable, so that you're able to tell, you're able to map the DNA. But it is not so with RNA, and I'll tell you the reason why mapping or sequencing RNA in PCR is difficult. I'll tell you in a minute. So he says, consequently, it cannot be used to diagnose infectious diseases. So he denied that PCR should be used for any diagnosis. Now let's go to the other challenge concerning RNA. The problem with RNA-based viruses is that the RNA itself is highly variable, so it changes with a slight perturbation. If it is disturbed any little, it is going to change. So this means that even if you are to take a sample from the body, that is from a living tissue, you move it away from the living tissue, and the sample is exposed to air, it is going to change. Even before you mix the sample with the reagents or you put it in the culture media, the sample is going to change. And it is changing because RNA is highly variable. So RNA exhibits high variability in concentration across different cells, and specific types like messenger RNA and long coding RNAs. For example, the types of RNAs we find in viruses, they are notoriously variable in sequence. Therefore, if you run it in PCR, most likely the product you get from the PCR is totally different from the sample that was taken from the body. So that is what we mean by high variability. So that is why you hear even mainstream science claiming that viruses have very high mutation rates. It is because of this RNA, because it's highly variable. Now, whether they have RNA or not, whether they have DNA or not, it's another issue. But for now, we want to challenge or to raise questions on the issue of maintaining RNA for 87 years. 87 years, from 1918 all the way to 2005. We want to confirm whether that position is tenable. Okay. So when we come to the technical issues now as to who discovered the virus, we have other questions that are coming up. If you are to type this question, on your search engine, who discovered the Spanish flu virus, you not get a single name being given. So they will say no one discovered the Spanish flu virus. But here we have a gentleman called Jeffrey TAUBENBERG who sequenced it, and yet it is said he did not discover the virus. Now, the truth of the matter is, there is a conventional practice in medicine. Anybody who makes a major discovery has to be awarded by the Nobel Peace Prize Committee in medicine or physiology. But Dr. Jeffrey TAUBENBERG, who sequenced, was not awarded the Nobel Peace Prize. And then you ask on your search engine like Google, why did he not receive the Nobel Peace Prize? And the answer you get is, he did not receive the Nobel Peace Prize because his contribution was sequencing the Spanish flu virus genome long after the original pandemic. That it took a very long time between the pandemic and the time he sequenced it. But this is not enough proof because, after all, he discovered it, if he discovered it. So they continue to say that the Nobel Peace Prize for the discovery of the flu virus itself was never awarded. So they acknowledge no one has ever been awarded for the discovery of the flu viruses. So you ask yourself, why have people not been awarded these prizes? So did we have the actual discovery, or this is just information that we need to criticize because we do not have anything else to back up that information? Okay, so this is the mystery surrounding the Spanish flu virus. So in summary, we are saying that it is unreasonable for a sample to have to store a virus for 87 years since the living organism died. And we know that viruses depend on living cells to replicate. That is, if you were to take what science is about, replication depending on living cells. So we had dead bodies for more than 80 years, but somebody was still able to take a sample. Then we also told that the virus was reconstructed. If it was reconstructed, meaning it was not the original. And this is where most people have based their trust in the information being given by mainstream science, that this is actually what caused the pandemic. Please note, it was a reconstructed particle. And then the other thing, that it will be difficult for an RNA-based virus to maintain its chemical structure for more than 80 years. RNA, we've noted that RNA is highly variable. It's unlikely that it could have retained its molecular structure for all those years. So that is how we expose the mystery surrounding the Spanish flu virus. Until next time, when we meet in another presentation, be blessed, continue to like, share, and subscribe. Thank you.